These are my notes. This is my audience. When most people think about scientific exploration, they think about exploring space or maybe the depths of the ocean. But one of the most important forms of scientific exploration is going on in the DNA that's inside of me and you right now. The purpose of this speech is to inform you of what genetic sequencing is, the possibilities unlocked by the genetic sequencing of the human genome, and the ethical questions that come with those possibilities. I believe I'm a credible source to talk about this material because I'm a biology major, so I interact with this material often, and I've also done extensive research to prepare for this speech. I believe that genetic sequencing will impact society because I see it as the cornerstone of the future of the sciences, especially one that affects society most, like medicine. Before I get into a description of my topic, I would like to start by explaining what a person's genetic sequence is. So according to Miguel Garcia Sancho in Genetic Information in the Age of DNA Sequencing, a person's genetic sequence is the precise linear structure, the sequence, of nucleotides that constitutes the DNA molecule. So we all know A's, T's, C's, and G's make up DNA. So if a person has A, C, T, G in that specific order, that would be their specific genetic sequence. I will further elaborate on that by starting a description of my topic, and I'm going to tell you what a person's genetic sequence does. So here we have a gene that's part of the chromosome and the specific DNA sequence that makes up that gene. So in this example, this DNA sequence starts with CAT, and let's just simplify it down to those three bases. So CAT in that specific order codes for a protein that makes pigments to turn this flower purple. So if the sequence was instead something like TCG, it would code for a different protein that might produce blue pigments that would make this flower blue. So the manipulation of a sequence changes the proteins that change expression. One of the biggest projects associated with genetic sequencing is the Human Genome Project, which was completed in 2003. And what it is an international project that decoded the human genome. So what it did was went through the entire DNA sequence of a human and found out which parts coded for what proteins that led to which expression. It was a pretty big project for geneticists. A lot of them equate this project to the man on the moon for space exploration. So it was a pretty big deal for them. And it also unlocked several possibilities. One of these possibilities is genetic engineering of the human genome. We have been doing genetic engineering for a long time now with things like GMOs, which would be genetically modifying plants to make them bigger and sweeter and more suitable to sell in the modern market. And we also use it in farming with a pH. According to John Maddock in the Australian Strategic Policy Institute, genetic engineering has its roots in the manipulation of genes and microbial hosts to produce pharmaceutical products such as insulin. So what they'll do is they will genetically engineer the sequence of these bacteria to amplify the production of insulin, and then take the insulin from the bacteria and use it to treat people with insulin deficiencies like diabetes and stuff. But following the complete sequencing of the human genome, we can now genetically engineer parts of the human genome like we do with bacteria. And that had led to a bunch of ethical questions about whether we should or could do that. But before we get into that, I'd like to establish a personal connection with this material. Like I mentioned previously, I'm a biology major at Henderson, so I interact with this material on a daily basis. Another connection I have to this material is that I could see this being a future career possibility for me because the science of genetics is a relatively young science and it's on the rise. So if that science is on the rise, that means job opportunities are on the rise. So I could see myself possibly getting into that in the future. And finally, I'm connected to this material in the same way that you all are connected to this material, and that its advancements in the medical field. So maybe when I'm getting older and I start, falling apart, genetic engineering could be the solution to some of that. So I'm related to it in that aspect as well. I feel like I have already established why I think that genetic engineering is important in the medical field. But just to reinforce that, I'd like to give two examples. And the first one is Nicholas Navin's delineating cancer evolution with single-cell sequencing. So this is a study that Nicholas Navin did, in which he developed a single-cell genome sequencing method for cells that lead to tumors. This single-cell genomic data could be used to reconstruct tumor evolution. So before that, it was kind of a mystery as to how normal cells transitioned into cancer cells that cause tumors. And so what Nicholas Navin did was found the genome of a cancer cell and compared it to the genome of a normal cell and spotted the differences. And maybe he'll approach those differences in the future, and that could help lead us to reducing the effects of cancer or curing it. Another example is in Samir Damani's Decoding Schizophrenia, in which he sequenced the exome, the protein coding portion of the genome, of 14 people with sporadic schizophrenia. And what he did was he took the genome of those people with schizophrenia and compared it to the genome of people without schizophrenia and looked for the differences in between, and maybe he'll approach those differences to reduce the effects of or cure schizophrenia. So, although I've mentioned a lot of examples that paint genetic engineering in a positive light, there's a lot of debate within the scientific community as to whether we should do any of this because of the safety of it. We don't know the effects of it as a person grows old, or the effects of genetic engineering on the future generations of that person that has been genetically engineered. There's also questions about the equity of it. A lot of people think that only the rich would be able, or it's more than likely that only the rich would be able to access technologies like this. So that kind of fuels the rich get richer idea that not a lot of people are with. In conclusion, the purpose of this speech was to inform you of what genetic sequencing is and the possibilities that are unlocked by it and the ethical questions that come with those possibilities. Thank you for listening to my speech.